students, welcome back. Today, again, we are dealing with class 10 from the chapter Life Processes. Here, we are learning nutrition in plants. Students, in the previous class, we learned about nutrition in animals, where the human digestive system we learned in detail, right? The animals, including the human beings, they undergo heterotrophic mode of nutrition. Also, we learned the importance of nutrition and the functions of food in our body. Dear students, as we are learning today about the nutrition in plants, we all know that the plants are also the living organisms. They too need food because they too perform many life processes in it, right? But the difference between us and the plants, we have different systems in our body to perform the various functions. But the plants don't have all the systems, right? But then too, the function should be carried out and the plant also needs food. But dear students, the plants, especially the green plants, can synthesize their own food with the help of simple raw materials like carbon dioxide and water with the help of sunlight. Of course, with the chlorophyll that is present in the leaves of plants. Here, during this photosynthesis process only, oxygen is released as a byproduct, which is very important for all the living organisms for the respiration process, right? Here, the energy breakdown takes place with the help of the digestive food. Today, we are dealing with autotrophic mode of nutrition in plants. The plants are also defined as the producers because of the photosynthesis process, and with this, they are supplying the food for all other organisms, including the non-green plants. Dear students, how can we represent the photosynthesis? So, as I told you, this takes place in green leaves of a plant, and the food is prepared here is in the form of simple sugar called glucose, and later this glucose is stored in the leaves as starch, right? But all this can take place only with the help of sunlight. Here is the equation, what we can see, which represents the photosynthesis, both word equation and symbolic representation. So the simple inorganic raw materials like carbon dioxide and water, in the presence of light and chlorophyll, the plants prepare glucose and oxygen is released. The symbolic representation is 6CO2 plus 6H2O, the presence of light and chlorophyll. The glucose, which is represented as C6H12O6 plus oxygen 6O2 is released into the atmosphere as a byproduct. Dear students, already we know the necessary conditions which is called the photosynthesis, right? Now, just now we learned that the starch is the final product which is stored in the leaves of plants. But how can we know that the starch is synthesized there? This we can know with a simple test called iron test. So this we deal with at the end of the topic. Students, here are the three steps which is involved in photosynthesis with two phases of photosynthesis, light reaction and dark reaction. Here, the first point is absorption of sunlight energy by chlorophyll. So chlorophyll helps in trapping of sunlight which is required for the photosynthesis process. So here, as light is involved in this case, this process also can be defined as photochemical reaction where light energy is converting into chemical energy. Also can be called as photolysis. Photo means light, where lysis means break. Okay. Yeah, the next step is conversion of light energy into chemical energy and splitting of water into hydrogen and oxygen with the help of light energy. The third step is the reduction of carbon dioxide by hydrogen to form carbohydrates like glucose, utilizing the chemical energy. So these are very important in the view of our examination. Dear students, let's now know how the plants obtain carbon dioxide from the atmosphere. Here, dear students, we have to recall from the class 9 what we have studied from tissue lesson about the stomata, the structure of stomata, the functions of stomata. At the same time, we also learned about the vascular tissues, the xylem and phloem and their functions. So all this you need to remember. Now, so the stomatas are the tiny pores which are present on the surface of leaves, which are guarded by guard cells. So these guard cells are the one which controls the entry of water and exit of water. Also, the stomata functions. It is helping in the exchange of gases, right? So when water flows into the guard cell, they swell. The guard cells swell, become curved, and cause the pore to open. On the other hand, when guard cells lose water, they shrink, become straight, and close the stomatal pore. Dear students, you can see the diagrammatic representation of the stomatal open and the stomatal closed. You can note the difference between it, right? On the other side, you can see the position of stomata in the lower epidermis of the leaves. So in between the upper and lower epidermis, there you can see the mesophyll cell, which has the chloroplast in it. Then, so that is how the carbon dioxide is absorbed by the stomata for the synthesis of food. So let us see now how the plants obtain water for photosynthesis. The water required by the plants for photosynthesis is absorbed by the roots of the plant from the soil through the process of osmosis. Again, from class 9. The osmosis can be defined as where it is helping in the movement of only the water molecules from its higher concentration region to its low concentration region to a semi-permeable membrane, right? The water absorbed by the roots of the plant is transported upward to the xylem vessels. Xylem vessels are helping in the transportation of water from the roots to the leaves. The plants are also in need of certain other raw materials here too, because during photosynthesis, it is only the carbohydrates which are synthesized. But other than this carbohydrate or glucose, the plants are also in need of nitrogen, phosphorus, iron, and magnesium. So all these are present in soil. So along with the water, these are also taken by the roots or absorbed by the roots. Students, you have also learned about nitrogen cycle and the importance of nitrogen for both plants and animals we have done. Because with the help of nitrogen only, the plants can synthesize its protein and animals. Then from the plants, it can take this protein and can synthesize their protein, animal protein. Then where exactly this photosynthesis takes place in plants? Of course, it is the chloroplasts. So what are the chloroplasts? Chloroplasts are the type of plastid. The plastid is an organelle which is existing only in a plant cell, right? So here, the site of photosynthesis in a cell of the leaf are chloroplasts, which contain a green pigment called chlorophyll. Chloroplasts are present in the photosynthetic cells, that is nothing but the mesophyll cells of green plants. And these cells contain more chlorophyll than other plant cells. Dear students, in the tender plants, you might have noticed the stem is also green in color, right? So, but here the chlorophyll is abundantly present in the leaves of plants. Here is the structure of chloroplast. Chloroplast is a double membrane structure with outer membrane and inner membrane filled with stroma within inner membrane. And here within stroma, where you can see the disc-shaped structures, which is called as grana. So this grana, many discs are piled one above the other. Together it is called as thylakoid. So each thylakoid is interconnected with stroma lamellae. Dear children, I told you about the two phases of photosynthesis. It is light and dark reaction. So the light reaction is taking place in the presence of light. That is during the daytime, where the conversion of light energy into chemical energy, and at the same time, water is split into its respective ions, that is hydrogen and oxygen. And during dark reaction, which is taking place in stroma. So here, the reduction of carbon dioxide to carbohydrates is taking place in the absence of light or in the little presence of light. Dear students, so this is now we are going to learn about in detail the three experiments which deal with that the sunlight is necessary for photosynthesis and chlorophyll is necessary for photosynthesis as well. The starch is synthesized during photosynthesis For all this, one basic test is there, that is iodine test. So here, before we get into the experiments, here we just have a look at how the iodine test is carried out. The leaf which has to be tested for all these experiments must be heated or boiled in water. After boiling in water, that has to be shifted to another test tube, where in this test tube we are going to take up alcohol. Then, direct heating of alcohol is not allowed as it is very dangerous. So, we are adopting double boiling or water bath. So, after boiling the leaf in alcohol, the leaf is taken out and is washed well in cold water. Then, after this leaf is transferred into a petri dish where few drops of iodine is dropped there. If the color changes to a blue-black, that represents the presence of starch there. Okay, let's get into the experiments here. Here is the experiment to show that chlorophyll is essential for photosynthesis. What is the procedure for this? Dear students, for all the three experiments, we are going to follow the common procedure here. So we need a potted plant with variegated leaves for this experiment. Variegated leaves. The example I can give you, the plant is croton. You might have noticed the leaf of a croton plant, which is of multiple colors, right? Many patches of different colors you can notice there, along with green patches as well. Students, this potted plant, we need to keep it uh, in a dark place for about 3 days to destarch its leaves. Now, keep the plant in bright sunlight after 3 to 4 hours. Then, pluck one of the variegated leaves from the plant. Boil it first in water for a few minutes and then in alcohol using a water bath so that it removes the chlorophyll from the leaf. Wash the decolorized leaf with water. Place the leaf in a petri dish and pour iodine solution over the leaf. After a few minutes, rinse off the excess iodine and absorb the leaf. What do you absorb? So wherever there is green patches, there and all we can see the color changed to blue-black, which represents the presence of starch. So from this we can know that, or we can prove that chlorophyll is necessary for photosynthesis. So other than the green patches, we cannot notice any color change here. Another experiment, this is to show that sunlight is necessary for photosynthesis. Here, again, we need a potted plant with green leaves and place it in a dark place for about 3 days to destarch its leaves. Then cover the part of the leaf with 2 uniform pieces of black paper and then fix the cover in position with 2 paper clips. So why are we covering the leaf with the black paper, children? Here, this black paper will not allow the sunlight to reach that particular part of the leaf. So only this is covered with a black paper. Then keep the plant in sunlight for a day. Remove the pieces of black paper from the leaf. Then go for iodine test. Destarch the leaf, boil it in alcohol to remove chlorophyll. So from this. Then test for the presence of starch in the leaf. Here is the diagrammatic representation for the experiment, as you can notice there. So here we can observe that the uncovered part of the leaf has turned blue-black. Uncovered part of the leaf has turned blue-black. This shows that starch is formed only in those parts of the leaf which was exposed to sunlight. And starch is not formed in the part of the leaf which was covered with a piece of black paper. So here from this experiment, we can know that sunlight is essential for photosynthesis. The last experiment here is to show that carbon dioxide is essential for photosynthesis. This is a procedure. So take a potted plant with a long leaves. So place it in a dark place for about 3 to 4 days to discharge its leaves. Then take a wide mouth bottle. So this is what we are going to do for this experiment. So we need a wide mouth bottle, which is we are going to take up some KOH solution, potassium hydroxide solution. So why the solution is used in this experiment? Because this solution absorbs carbon dioxide gas. Okay. So only in the bottle we are going to take up the solution of potassium hydroxide, and with the help of a slitted rubber cork, we are going to insert one of the leaves into this. So we need to we need to make the bottle airtight and place the operators in bright sunlight for few hours. Now, detach the leaf and test for the presence of starch in it. Here is the diagrammatic representation. So here is a diagrammatic representation for the experiment. You can see the bottle there, it is fixed with the rubber, splitted rubber cup, and the leaf is inserted to the splitted cup. Airwood solution, which is to absorb the carbon dioxide which is present in the bottle there. So when we go for iodine test, so here the leaf part which is outside the bottle will only show the color change, so which represents that the starch is present only in that part. So the leaf part which is inside the bottle will not show any color change. So this represents that, this proves that carbon dioxide is also essential for the photosynthesis process. So that's all, students. Thank you.